I am Professor Joshua Sharp of the University of Mississippi School of Pharmacy, Department of Biomolecular Sciences. This is another video in our series on hydroxyl radical protein footprinting and fast photochemical oxidation of proteins. In today's video, we will be talking about reproducibility and robustness in FPOP experiments, giving you an introduction to questions of radical dosimetry, radical scavenging, and how best to perform your experiments in ways that not only can you reproduce the results in your own lab, but also allow other labs to accurately reproduce the results that you find. This video series is made possible through a grant from the National Science Foundation, and my personal financial conflict of interest statement is shown here in the bottom right. As always, if you have any questions or recommendations or requests for new videos, please feel free to contact me by email, jsharp@olemiss.edu. So if you are new to FPOP, as far as how the experiment works, what kind of information it can give you, I would strongly recommend that you stop this video and go find our introduction video to FPOP and hydroxyl radical protein footprinting. That's really the best place to start off this series. If you don't watch that first, chances are you will be a bit lost in this video. That video, as well as other videos in our series, can be found either at our YouTube channel for the Sharp Lab, or at our web portal for the lab at masspec.olemiss.edu/videos. But let's start by taking a look at a typical FPOP experimental workflow. So in this case, we have a protein, adalimumab biosimilar. So this is a biosimilar for a pharmaceutical and monoclonal antibody, and we want to understand how two different formulations affect the conformation and aggregation of this protein. So we want to test the chain. We want to test basically what happens when you change the buffer. So we perform FPOP of the protein under two different buffer conditions. In one condition, we have 50 millimolar sodium phosphate buffer at pH 6, and in the other condition, we have the same amount of protein, 50 millimolar sodium citrate buffer at pH 6. We perform the same experiment under identical optical conditions, so 100 millijoules per pulse, 100 millimolar hydrogen peroxide. Everything is the same as far as the FPOP. The only difference is sodium phosphate buffer versus sodium citrate buffer. When we perform this experiment, we measure the amount of oxidation of various peptides on the adalimumab biosimilar. What we see is that the sodium citrate sample shows considerably less oxidation all across the protein. Normally, this is interpreted as a protection of that region of the protein from the solvent, which would suggest something along the lines of an aggregation event that's protecting the vast majority of this protein. However, what I want to point out to you today is that's not actually what's going on here. And in order to explain what's actually happening in this particular case, I need to give you a little bit more of a detailed look of how the hydroxyl radical protein footprinting experiment works at the molecular level. So let's take a closer look. So here we have our UV krypton fluorine excimer laser and our hydrogen peroxide. And what we're going to do is we're going to fire a laser pulse at the sample. The hydrogen peroxide is going to absorb a photon of the UV light, which will cause the hydrogen peroxide to photolyze into two hydroxyl radicals. Now, of course, we're not firing one photon at one molecule of hydrogen peroxide. We're firing many photons at many molecules of hydrogen peroxide, so we are going to make many hydroxyl radicals. We're doing all of this in the presence of our analyte protein, the adalimumab biosimilar. And what will then happen is the hydroxyl radicals that we make from the hydrogen peroxide, from the photolysis of the hydrogen peroxide, will diffuse through the aqueous solution. And when it reaches the surface of our target antibody, it will react with amino acid side chains and form an oxidized amino acid. And of course, all of the hydroxyl radicals will all diffuse to the surface. And in addition to the hydroxyl radicals, you also have molecular oxygen, which takes part in the chemistry that's going on here. But the upshot being that in most cases, for most amino acid side chains, the major product is going to be incorporation of an oxygen atom. Whether the oxygen atom comes from the hydroxyl radical or whether it comes from ultimately from the molecular oxygen, you will get the addition of an oxygen atom to your amino acid side chain. This occurs on the surface of the protein, and this is how we use hydroxyl radical protein footprinting and FPOP to map the surfaces of proteins. However, you'll notice not all of the hydroxyl radical diffuse to the surface of the protein. That's because hydroxyl radicals are a very, very powerful oxidant, and they're not specific to proteins. They will oxidize lots and lots of things. And so you have all of these reactions that are competing with one another. So one reaction that's always present in any hydroxyl radical protein footprinting experiment is the reaction of the hydroxyl radical with another hydroxyl radical, in this case to recombine and reform the hydrogen peroxide. This is a very favorable reaction, and so it is always occurring in competition with the reaction of the hydroxyl radicals with your analyte protein. So let's take a look now at what happens if we now add in a new player, in this case an organic buffer. So you still have the hydroxyl radical that diffuses to the surface of your protein. But in addition, there's now a new reaction pathway for the hydroxyl radicals. They can react with one another to recombine. They can react with the amino acids on the surface of your protein to oxidize the protein. But they can also diffuse to the organic buffer and react with the organic buffer, oxidizing the buffer molecules. Now, we are not changing the optical output of our laser. We're not changing the concentration of the hydrogen peroxide. So we're making the same initial concentration of radicals. But we added in a new pathway for consuming these radicals. So the upshot of that is that in the end, you get fewer radicals that actually make it to the surface of your protein to oxidize your protein. And so when you compare your protein in sodium phosphate buffer, which is largely unreactive to hydroxyl radicals, versus your protein in an organic buffer, which is reactive to hydroxyl radicals, you will see considerably fewer oxidations on the surface of your protein in the organic buffer, even though the conformation of the protein has not changed. This effect is known as hydroxyl radical scavenging. You have a new molecule in solution that is competing with your analyte for the hydroxyl radical, therefore reducing the amount of oxidized analyte protein that's formed without changing the structure of the protein. Since hydroxyl radicals will react with a wide variety of things, this radical scavenging is always an issue that needs to concern the experimenter in hydroxyl radical protein footprinting. And the method for measuring and correcting for hydroxyl radical scavenging is not, it has been with us for most of the lifetime of the hydroxyl radical protein footprinting method itself. This technology is known as hydroxyl radical dosimetry, a method for measuring the effective amount of radical and the scavenging capacity of your buffer or of your system. And there are many methods that have been published for doing radical dosimetry in hydroxyl radical protein footprinting measurements. The first one was published for specifically for doing hydroxyl radical protein footprinting by water radiolysis in an X-ray synchrotron beamline, published by the Chance Group in 2007. But there have also been methods published specifically for FPOP. The Gross Lab published an isotope dilution GCMS method. And then my lab published an internal reporter method, both in 2015, internal peptide reporter method. This internal peptide reporter was also published by the Gross Group in 2017. And then the method I'm going to spend most of my time talking about today for doing radical dosimetry is a method that we published in 2015 known as adenine UV absorbance radical dosimetry. And we've published several papers. Our initial publication, 2015, a method adapting this from FPOP to microtiter plate photolysis. And then finally, a real-time method for doing adenine dosimetry, which I will highlight today, published in 2018. All of these methods have the same basic idea behind them. In all of these methods, you are adding in an exogenous molecule to your solution that provides a competing pathway, reaction pathway for the hydroxyl radicals that you make. However, this particular molecule has a particular way that you can measure the amount of oxidation of this molecule. And by measuring the amount of oxidation of the molecule, you can understand more about how much radical you made and how much radical is being consumed by other pathways. Adenine radical dosimetry is particularly attractive for one very, very simple reason, which is the ease at which you can actually read out the oxidation. So this is adenine, and adenine has a UV absorbance spectrum that looks like this, with a strong absorbance band at about 265 nanometers. Now, if you add in hydrogen peroxide, that absorbance band stays at 265 nanometers. If you expose the adenine to a KrF excimer laser, that absorbance stays at 265 nanometers. However, if you expose the adenine to both hydrogen peroxide and the excimer laser, so you expose it to hydroxyl radicals, the adenine becomes oxidized, and the oxidized adenine loses that strong absorbance band at 265 nanometers. So you can see a big drop off in UV absorbance at 265 nanometers when the adenine gets oxidized. This allows us to actually measure the oxidation of our internal standards simply by UV spectrophotometry, which is a very simple readout. So let's take a look at what this, at how this works at the molecular level. Again, we have our excimer laser firing our photons at hydrogen peroxide, making this initial burst of hydroxyl radicals in the presence of our analyte protein, molecular oxygen. But now we've added in this organic molecule, our adenine. The hydroxyl radicals will diffuse both to the surface of the protein and to our adenine dosimeter, and it will oxidize. What we can then do is measure the UV absorbance of our adenine dosimeter. And we can measure it before we generate the hydroxyl radicals, and then measure it after we've generated the hydroxyl radicals, and measure the change in absorbance at 265 nanometers. And this change in absorbance is going to reflect the proportion of the adenine that has become oxidized by this burst of hydroxyl radicals. So in this particular case, using this particular spectrophotometer, we get a change of 8 milliabsorbance units. Now, if we repeat this experiment, except we add in a radical scavenger, so in this case an organic buffer, we make the same initial burst of hydroxyl radicals. Now it has 3 reaction pathways in addition to recombination. It can react with our analyte, it can react with our adenine dosimeter, or it can react with our organic buffer. Since you have an additional reaction pathway competing for the same initial pool of hydroxyl radicals, the amount of oxidation of the adenine and of the protein will be reduced. And since we can easily measure the amount of oxidation of the adenine by UV spectrophotometry, we can see our change in adenine absorbance has gone from 8 milliabsorbance units before, in the absence of the organic buffer, now to 5 milliabsorbance units in the presence of the organic buffer. So we can tell that we have increased the radical scavenging capacity of our buffer, and we can expect that our protein will have fewer oxidations on the surface. So now you hopefully understand how radical dosimetry can help you determine when you have differential labeling due to differential radical scavenging capacities. But how do we correct for this in this experiment? So for example, in my adalimumab experiment, I have the protein and sodium phosphate buffer, I have the protein and citrate buffer. I know that the different radical scavenging capabilities of the citrate are going to impact my footprint on the protein. How do I correct for that? The traditional way would be to take my protein and sodium phosphate buffer and add in a radical scavenger until I match the scavenging capacity of the citrate buffer. However, as you can see here in the oxidized protein, when I have a higher radical scavenging background for the same amount of radical, I get fewer oxidations on the surface of my protein. And in hydroxyl radical protein footprinting, oxidations equal information. The more oxidation you can get within the appropriate time scale, the more information you have on the structure of your protein. So if you have to increase the scavenging capacity of your buffer, you're reducing the amount of information. Is there a way that I can preserve the amount of information that I'm generating? In principle, the answer is yes. And here's how you would do it. Simply increase the amount of hydroxyl radicals you make, either by turning up the UV fluence of your laser, so the number of photons that you are pumping into the same amount of hydrogen peroxide, or increasing the concentration of hydrogen peroxide in solution. Either way, you will make more hydroxyl radicals. These hydroxyl radicals will still diffuse around. They will still be split among all the different reaction pathways. But by measuring the change in absorbance in your radical dosimeter, you can tailor the amount of radical you make until the readout on your dosimeter is identical to that of your initial conditions. So for example, the sodium phosphate buffer with fewer radicals that are generated. Now we have the organic buffer, but we generate more radicals, and we keep generating more radicals until we reach that identical dosimeter reading. Okay, a change of 8 milliabsorbance units at 265 nanometers. Once we've done that, we know we've oxidized the adenine the same amount under these new conditions. So we should have oxidized the protein surface the same amount if the protein has the same conformation. So this process is known as compensation. We are compensating for the differential radical scavenging capacities of different samples, with the idea being that if we compensate based on the radical dosimetry readouts, then we should get identical footprints for identical conformations, and any change in the footprint would reflect a change in the conformation of the analyte. So just to give you an idea from a practical standpoint, since this is a tutorial video, a practical standpoint of how this works, how do you actually compensate in these experiments? This is a picture of our optical bench, which is a fairly simple example. There are more simple optical benches you can make, but this one is a relatively simple example that will work and allow you to do the experiments that we have pointed out here. Our laser is off screen over here to the right, firing the beam from right to left. Then we have our lens that focuses the excimer laser beam. Here we have our syringe pump, pumping our sample through our fused silica capillary. We have two capillary holders held in place by these optical posts. Their job is basically to hold the capillary firmly and rigidly in the path of the focused beam to make sure it doesn't wobble around. So to do that, we have these compression sleeves inside of these stainless steel fittings screwed into the capillary holders. These gently pinch the capillary and hold it in place firmly in between, in the path of the beam between these two capillary holders. Once it leaves the capillary holder, here we have our capillary UV spectrophotometer that measures our adenine absorbance, and I'll talk more about that in the next slide. But you'll notice we have one other element here, this aperture. What does the aperture do? Well, if you'll think back to our previous slide, when I told you about how we actually compensate to make more radical, I told you what we do is you can increase the fluence of your UV laser. So for KRF excimer lasers, the way you increase the energy of the laser is you turn up the voltage on the thyrotron. This increases the energy per pulse that's being emitted from the laser. When you do that, however, in an excimer laser, you don't just increase the fluence or the photon density per unit area of the beam cross-section. You also increase the beam size. So this is a beam profile of our excimer laser at 80 millijoules per pulse. versus 140 millijoules per pulse. And what you can see is the beam size changes considerably as we increase the energy. It gets much, much larger. Now, if you'll think back to our introduction to FPOP, you'll remember that in FPOP, we carefully measure the profile of the beam and then time the flow rate of our solution through the capillary in order to ensure that each and every volume of sample is only illuminated by a single laser shot, with a exclusion volume or an unirradiated volume in between each laser pulse. Well, if our laser spot is changing size, then that means the illuminated area of the capillary is changing in width, which means that if we want to maintain our exclusion volume, we have to adjust our flow rate. That means that anytime you change the beam, you have to reprofile the beam cross-section, recalculate your flow rate. It becomes very, very easy to accidentally eat into your exclusion volume or change your exclusion volume, or even in some cases double illuminate part of your sample, which, if you listen to our introduction to FPOP video, you know is a big no-no in this experiment. It's a good way to get bad results. So what does the aperture do? The aperture ensures that no matter how large your beam gets, the only part of your beam that impinges on the capillary is the part that's limited by this 1 millimeter hole inside of this metal or ceramic disc. So if the beam expands in size, it's okay, because only 1 millimeter diameter of that is going to make it through the aperture and impinge on your capillary. So you don't have to change your flow rate. You don't have to worry about eating into your exclusion volume. The aperture also helps take care of what's known as laser jitter or axial diffusion of the laser. So laser can actually change its trajectory coming out of the beam. It's not by much, it's only by a degree or a fraction of a degree. But since we're having to measure this illuminated cross-section very carefully, a deviation of a degree can actually make quite a difference. So with the aperture, even if the laser drifts a little bit left or right from shot to shot, the amount of the laser that makes it through the aperture remains stable and dictated by this aperture. So this now allows us to either turn up our laser output or even change the focal plane of our lens to change the fluence of our laser at the capillary and compensate, increase, and make more or less hydroxyl radicals without having to recalculate our flow rate through the syringe pump. So this is what the experiment actually looks like. Here on the left, I've got the readout from our inline spectrophotometer. And the inline spectrophotometer is actually fairly simple in construction. You have your capillary, your photolysis capillary, and it's passing through a optical stage that has a 265 nanometer UV LED. This light is focused and guided by a series of ball lenses to be focused into the lumen of your capillary, and then directed onto a UV photodetector. And all you're measuring is the change in the light that impinges on the photodetector based on the change in absorbance from whatever is in your capillary. So this is the trace, the UV absorbance spectrum, and as you can see, you have a UV absorbance, and then as soon as I turn on my laser, this absorbance drops. Now in this particular experiment, I'm trying to compensate for the presence of additional hydroxyl radical scavenger. So I know in my initial experiment that I'm trying to match, my radical dosimeter gave me a change in adenine absorbance from 79 milliabsorbance units to 66 milliabsorbance units here to the dotted line. When I turn on my laser under the same conditions in the presence of the radical scavenger, however, I only drop from 79 MAU down to about 70 MAU. So what I can then do is increase my laser output energy and watch in real time as this falls, as our adenine radical dosimetry falls, until I reach our normalization target, in which case I know that I've reached the proper laser energy. I can stop there and start collecting my sample, and I'm now under compensated conditions. I'm now creating the same effective radical concentration for that my dosimeter sees, and therefore that my protein analyte sees. Since dosimetry readings correlate linearly with peptide and protein oxidation, this should mean that as long as I compensate based on dosimetry, I should be able to get the same footprint for the same structure under two widely different radical scavenging backgrounds. But does it actually work? Here is where we demonstrate that yes, this technique actually works. So we took a protein, simple standard, in this case myoglobin, and we performed an FPOP experiment. In blue, it shows the results of the experiment we performed in sodium phosphate buffer under these optical conditions, so 11.66 millijoules per square millimeter per pulse. And in blue, you can see the amount of oxidation of each peptide we detected in myoglobin. Now, if we then change the 10 millimolar sodium phosphate buffer to 10 millimolar MES buffer, which is an organic buffer, a radical scavenger, we perform the experiment under the same optical conditions. We get very different readouts from our adenine dosimetry. The adenine dosimetry goes from a delta absorbance of 19 to a delta absorbance of 7. And what you see in the footprint is a noticeable reduction in oxidation for every peptide. If you don't do the radical dosimetry, this would look like the entire protein's being protected, and so you have a big aggregation event or some kind of structural collapse. But now you and I know better. Because we saw the change in the adenine dosimetry, we know that this reduction is not due to a structural event, it's due to changing the radical scavenging capacities of our solution, in this case by changing the buffer. So now can we compensate for this new higher radical scavenging capacity? So we perform the same experiment in 10 millimolar MES buffer, but now instead of keeping the same optical conditions, we increase our laser fluence until we reach the same dosimetry conditions. All right, within error, the dosimetry readings are within error of one another. But to do that, we had to go from 11.66 millijoules per square millimeter to 18.75 millijoules per square millimeter. And when we do that and take the radical footprint of myoglobin, we see that it is now identical for every peptide. We have successfully compensated for widely different radical scavenging capacities, and we've received the exact same footprint from both samples. Since all of our biophysical data indicate that myoglobin has the same conformation in sodium phosphate buffer versus MES buffer at the same pH, then this is telling us we are getting the same footprint for the same structure under widely varying radical scavenging capacities. So this does work, and this allows you to preserve the amount of information that you get from your higher radical doses. And if you use the inline adenine dosimetry method, you can actually make this adjustment in real time. So let's go back to our adalimumab biosimilar, and let's apply our compensation technology to that. So if we look at the adenine absorbance in the sodium phosphate buffer, our adenine dosimetry gives us a reading of 9.767 delta absorbance at 265. If we perform the same experiment under the same optical conditions in citrate buffer, it now drops to 5.0. So a huge decrease in the effective radical concentration due to the scavenging capacity of the citrate. So now if we turn up our laser fluence, we make more radical, we can now achieve compensated conditions in the same citrate buffer where our delta absorbance 265 is statistically identical with that of our phosphate buffer. And we can measure our adalimumab response, our footprint. And what we see is we still see a protection in many areas of the protein in citrate buffer versus phosphate buffer. It's not as pronounced in the citrate buffer, and most of the heavy protection is limited to the tips of the FAB and the very, very end of the FC region, but we still see slight widespread protection. So is there a problem with our compensation? No. In this particular case, citrate buffer does cause the protein to aggregate, as we can tell from our dynamic light scattering data. In phosphate buffer, we have 28.7% of our dynamic light scattering intensity in the antibody monomer, and 71.3% of our DLS intensity in this large aggregate. When we change to citrate buffer, that equilibrium shifts up to so we have less of the monomer and more of the aggregate by about 7% of the DLS intensity. So what we're observing in this particular case is we're observing the protection that we get from the aggregation event itself. And based on the magnitude of the change, we can start talking about which regions are more or less involved in this aggregation interface. So we can detect the aggregation. We can also tell you something about the structures that are involved and the interfaces that are involved in the aggregation process. Now that illustrates radical scavenging and radical dosimetry and compensation in questions of changing buffers or changing formulations for pharmaceutical proteins. But does this, is that all that this is useful for, all it's important for? And the answer is no. Remember, hydroxyl radicals are incredibly strong oxidants. Almost any organic molecule is going to scavenge your hydroxyl radical to some extent or another. And this includes protein binding partners, and it includes organic ligands. So in this particular case, we're looking at a protein-protein interaction. We have a cation-independent mannose-6-phosphate receptor, CIMPR. This is a protein that binds to these mannose-6-phosphate glycosylated proteins, which are important for shuttling proteins to endosomes and maturation of endosomes into lysosomes. And then we have one of its ligands, which is a mannose-6-phosphate glycosylated protein known as PPT1. We perform the hydroxyl radical footprinting of the CIMPR domain 1 through 5 in the absence and then in the presence of its ligand because we want to see where does the mannose-6-phosphate bind to the CIMPR. And when we look at our radical dosimetry, we very quickly see that there's a problem. If we just look at the CIMPR domain 1 through 5, we get a dosimetry reading of 130 milliabsorbance units. But when we add in our protein binding partner, PPT, this shifts from 130 milliabsorbance units now to a change of only 73 milliabsorbance units. So we are scavenging an immense amount of our hydroxyl radical, even though we're doing this in the same buffer. Why? Well, remember, as far as CIMPR is concerned, PPT1 is not just a protein binding partner, it's a radical scavenger. It is now competing with your analyte protein for the same pool of radicals, and so fewer radicals are making it to the surface of CIMPR. So if we were to compare this footprint with this footprint, what we would quickly see is it would look like everything on CIMPR 1 through 5 is protected, and we wouldn't be able to tell anything structurally about the protein simply because, not because the PPT1 is protecting the surface of the protein from the solvent, but because the PPT1 is competing for the radical. It's scavenging the radical out of solution. So just like compensating for radical scavengers that don't interact with the protein, like buffers, we have to compensate for radical scavenging of the protein binding partner. So we increase our UV fluence until we get comparable dosimetry readings that are within error of one another, and we measure our footprint. And what we see now is rather than seeing protection at every single peptide, we have 1, 2, 3, 4, 5 peptides that are protected and 1, 2 peptides that are actually exposed. So we can actually see specifically which peptides show a change in their solvent accessibility and their protection from the solvent. And if we then take this information and map it onto the structure of the mannose-6-phosphate receptor, we can actually start to make sense of the data. So this is the structure of CIMPR, and shown in the red mesh are the regions that are significantly protected under compensated conditions upon binding of PPT1. And what you can see is these protected regions cluster to 2 areas, here and here. These are actually the 2 sites of mannose-6-phosphate binding for MPR. One mannose-6-phosphate binding region is in domain 3, shown in green, but it also requires domain 1 in order to be a high affinity site. So this is the high affinity mannose-6-phosphate binding site at the interface between domain 3 and domain 1. And then you have a low affinity binding site here in domain 5, which is also protected upon addition of PPT1. You'll also see we've got these green meshes here, as long as a little bit of protected region. The green mesh represents areas that become exposed upon PPT1 binding. And that indicates that there's actually a conformational change that's occurring here in this part of domain 4. So we can see the binding effects, and we can also see the allosteric effects of binding using the compensated hydroxyl radical protein footprinting. And remember, if you don't use your radical dosimetry to compensate for the radical scavenging of your protein binding partner, this whole protein is going to look protected. So in conclusion for today's video, I want to really point out that you have to use radical dosimetry if you want to have reproducibility, if you want to have robustness, if you want to be able to control your experiment and understand how it works. You have to do radical dosimetry. Always keep in mind, FPOP is not a spectrophotometric measurement. It is a photochemical measurement. And so in order to understand what's going on, you need to measure the photochemistry, not just the performance of your optical bench. There are lots of reasons for this. The first is questions of radical scavenging, which we spent a lot of time talking about today, but those are not the only things that can cause a change in your effective radical concentration. Hydrogen peroxide is a perishable laboratory reagent. It degrades fairly steadily and not incredibly slowly. And it's not always straightforward for you or your lab mates to always make sure that the peroxide you're using is in the same condition as it was for all of your previous experiments. And if you're using peroxide that's partially degraded, you're going to get a lower radical yield under the same optical conditions and nominal peroxide concentrations. Additionally, your optical path. You have to remember, if you remember looking back at our optical bench, this isn't a highly engineered, carefully graded optical bench, right? This is something that we have assembled ourselves. These are not high quality, delicately engineered photochemical cells. We're using a piece of fused silica that we've burned the coating off of, okay? And every time that we use a different piece of fused silica, we're changing the optical properties of our optical bench, because the fused silica itself can diffract the light. It can even in some cases focus the light. And different pieces of fused silica are going to have slightly different properties because fused silica is not made to do this accurately and reproducibly. All of these differences will impact the amount of hydroxyl radical that you make and that you deliver to your analyte. All of these effects can be measured using radical dosimetry. So it's important that you use radical dosimetry, and it's important that you report the radical dosimetry results for your experiments in sufficient detail for others to come along and reproduce your experiment. Even if your experiment shows no differential scavenging effects between two samples, if you want someone else to be able to reproduce your results, they need to be able to reproduce your effective radical yield. And they can't do that if all you give them are your optical path measurements. You need to give them your radical dosimetry measurements. And then finally, I hope I convinced you that dosimetry-based compensation works. You don't have to throw away data in order to compensate for radical scavenging effects. If you use your radical dosimeter to monitor the effective radical dose that you're delivering to your radical dosimeter, you can normalize that with the effective radical dose you are delivering to your analyte and just make more radical until your radical dosimetry reading is the same in both conditions. As long as you do that, and as long as you're working with a validated radical dosimetry system that is validated under the conditions that you're using, you're going to be fine. Thank you for watching this video. I hope it was educational. If you enjoyed this video, we are making several more, so please check those out at our YouTube channel or at masspec.olemiss.edu/videos. Again, if you have questions about this video or recommendations of other videos in this series that you would like to see, questions about how hydroxyl radical protein footprinting works, or some of the practical aspects that you would like us to make a video about, please feel free to email me. Once again, I'd like to thank the National Science Foundation for funding this series of tutorial videos, and I hope to see you next time for our next installment in the series. Thank you.